Welcome to this lesson on chromatin. Chromatin is a fascinating structure that represents uncondensed DNA combined with proteins. This is the form in which your genetic material exists for most of the cell's life cycle. Understanding chromatin is essential to grasp how cells manage and protect their DNA while still allowing access to genes when needed. Let's start by defining chromatin. Chromatin is a complex mixture of DNA and proteins found inside the nucleus of eukaryotic cells. It exists primarily during interphase, which is the longest phase of the cell cycle when the cell is not dividing. During this time, the DNA needs to be accessible for important processes like reading genes and copying DNA, so it remains in this relaxed, uncondensed form called chromatin. Chromatin has two main components. First is DNA, which carries all your genetic information. In chromatin, this DNA exists in a relaxed, extended form rather than tightly coiled. Second are histones, which are special structural proteins that provide support. These histone proteins act like spools around which DNA wraps, helping to organize and package the long DNA molecules efficiently within the small space of the nucleus. Now let's look at how chromatin is structured. DNA wraps around groups of 8 histone proteins, which we call histone octamers. Each unit where DNA wraps around histones is called a nucleosome, and these nucleosomes are the basic repeating units of chromatin. When you look at chromatin under a microscope, it resembles beads on a string, where the beads are nucleosomes and the string is the connecting DNA between them. Chromatin exists in two different forms. The first type is euchromatin, which is loosely packed chromatin. This loose packing makes it transcriptionally active, meaning genes in these regions can be read and expressed. The second type is heterochromatin, which is tightly packed chromatin. This tight packing makes it transcriptionally inactive, so genes in these regions are essentially turned off and cannot be accessed for expression. Chromatin serves several critical functions in the cell. First, it packages DNA into a more compact form so that meters of DNA can fit inside the tiny nucleus. Second, it regulates gene expression by controlling which genes are accessible and which are not. Third, it facilitates DNA replication by organizing DNA in a way that replication machinery can access it. Finally, chromatin protects DNA from damage by wrapping it around proteins and keeping it organized rather than loose and exposed. During interphase, DNA exists in its least condensed form as chromatin. This is crucial because the cell needs access to DNA for transcription, which is reading genes to make proteins, and for replication, which is copying DNA before cell division. The cell actually spends most of its life in interphase, so chromatin is the primary state of DNA organization for the majority of a cell's existence. Here's the key takeaway from this lesson. Chromatin equals relaxed DNA. This relaxed state is what allows all the essential cellular processes to occur during interphase. When DNA is in the form of chromatin, enzymes and proteins can access the genetic information to read genes, copy DNA, and perform repairs. Without chromatin's organized yet accessible structure, cells couldn't function properly.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.